doing today. Can you hear me? All right, great. I'm just double checking if you can see the two tablets on here on the screen. All right, so I did receive a couple of emails that had difficulty with completing the quiz. They were not aware of the due date of the quizzes. So what I will do is I will open all the quizzes for anybody who did miss any of the quizzes. You can get a chance to get them completed before the end of the semester. So I will be opening all the quizzes for you to be able to get them completed. All right, so today I will be opening all the quizzes to be due on the Monday before the end of the semester. All right, so I will be putting an announcement on Canvas as well for this. All right, so if you've missed any of the previous quizzes, regardless of what quiz you've missed, whether it's unit 1, 2, or 3, you should be able to get them completed. Please be aware that if you've missed the due date at the end of the semester, I won't be able to help because I would have to be posting the final grades for the course and I won't be able to have any delays after that day. All right. All right, so today we are starting in our last quarter, unit 4. And in this unit, we are discussing the reproduction and the digestion and metabolism. So for the reproduction is going to be our first chapter that we're going to be starting with. Reproductive system is going to be highly important for our upcoming lab exam. It's going to be representing around 60 or 70% of the upcoming lab exam. I will tell you exactly when the lab exams are going to be conducted and how you're going to be able to complete the proctored lab exam after break, so after everybody comes in. All right, so again, reproductive system is going to be representing 60 to 70% of the upcoming lab exam. So we're going to be starting with it and we're going to be discussing the digestion and metabolism on another class, on another lecture. All right, so for the reproductive system, what we're going to be discussing first, we're going to be learning the structures and functions of the male and the female reproductive systems. We're going to be understanding the gametogenesis, which is the formation of the sperms and the eggs. We're going to discuss the hormonal regulation of the reproductive function. So what are the hormones that are effective in regulating spermatogenesis in males and oogenesis in females? The menstrual cycles, the ovarian cycles, all those going to be discussed over this chapter. We're going to also be discussing some of the homeostatic imbalances that are going to be impacting the reproductive system towards the end of the chapter. So let's start first with the structure of the reproductive system. So the reproductive system has two major components, either in males or females. The first component is going to be the primary sex organs. And those are the gonads. In males, this is going to be the testes. And in females, those going to be the ovaries. What makes the gonads so special? Because simply they are the only part in your body that have the ability to produce gametes. And the gametes are, in males, the sperms, and in females, those gonna be the mature oocytes or the egg. So what is so special about the sperms and the oocytes? Simply, they have only half of the genetic code, half the number of chromosomes. We call those are the haploid cells. It's a haploid cell. What I mean by haploid means it contains only half of the number of chromosomes compared to the rest of the body cells, which we call diploid cells. This is going to be the first and major function here of the gonads, is to produce the sex cells or the gametes. Another function for the gonads will be to produce the sex hormones, which are the androgens in males and the estrogen and progesterone in females. The other component of any reproductive system is going to be the accessory organs, accessory reproductive organs. And the accessory reproductive organs are going to be like, for example, in males, the glands, which are the prostate gland, seminal vesicle, and so on. So you have glands associated. We have ducts through which the sperm is going to be traveling and in which the sperm is going to be produced. Erectile tissue, like the erectile tissue in the penis, all those are accessory organs. For females, we've got the uterus, we've got the fallopian tubes or the uterine tubes, we've got the vagina, we've got the clitoris. So all those are going to be accessory organs that will aid the gonads to allow reproduction to take place. But the primary sex organ, this is the one that's going to be of great importance because it is simply going to be producing the gametes, sperms and eggs. Starting first with the structure of the male reproductive system. We've got the gonad in here. This is the testis. The testis is pending down in a skin pouch. We call this skin pouch is the scrotum. Located just above the testis, we've got a set of ducts or tubules located above the testis. This is my epididymis. So if we're looking on here, this is going to be the epididymis. The epididymis is going to be conducting the sperms coming from the testis up through a duct that we call this duct on here is what we call the vas deferens or the ductus deferens. So again, we started by the testis on here. Sperms are going to be conducted from the testis to the epididymis, and then up from the epididymis through a duct. You see on here, this duct is going to be traveling up from the scrotum back to the pelvis. All this is what we call the vas deferens or the ductus deferens. So again, we've got here the testis located in the scrotum. The epididymis is located above it. The epididymis is going to be carrying the sperms up towards the pelvis through a duct called the vas deferens. It goes all the way to the pelvis. It's going to be traveling up to meet with a gland. We call this gland is my seminal vesicle. Seminal vesicle is located below the urinary bladder. The duct of the vas deferens, the ampulla of the vas deferens, along with the duct of the seminal gland, they will fuse together and they will form a common duct that will be traveling inside the prostate gland, which surrounds the neck of the urinary bladder and the proximal part of the urethra. So if I'm drawing here, this is the urinary bladder. The lowermost part of the urinary bladder, as you remember, we call this is the bladder neck, is surrounded by a gland, and we call this gland is the prostate gland, like this. Inside the prostate gland, you've got the seminal vesicle duct fusing with the ampulla of the vas deferens, forming another duct that we call the ejaculatory duct. So how many ejaculatory ducts do you think you have, a male reproductive system will have? One. How many ejaculatory ducts? One. Any other ideas? Two, three. So how many ejaculatory ducts is simply going to be determined. You see on here how many testes a person normally have. 2, right? Each one has its epididymis. Each epididymis is going to be conducting the sperms up through a vas difference. So how many vas difference do we have? How many ductus difference do we have? We've got 2. And each of them gonna be fusing with the duct of the seminal vesicle. So you've got 2 seminal vesicles, each of which will be fusing with the vas difference. So you've got 2 ejaculatory ducts. So you've got right ejaculatory duct, left ejaculatory ducts that will be releasing the semen to the proximal part of the urethra. This takes place where again? Inside which gland? What gland are we located now in? Can you remind me, please? What gland am I located in here? I am located in the prostate gland, exactly. So inside the prostate gland that surrounds the neck of the bladder and the proximal part of the urethra, the 2 ejaculatory ducts gonna be releasing the semen to travel through the proximal part of the urethra. If you remember, what did we call the part of the urethra that travels in the prostate gland? The part of the urethra that travels in the prostate gland. This portion of the urethra on here. Prostatic urethra, exactly. And again, it travels inside glands surrounding the bladder neck as well. It's going to be the prostate gland. The urethra keeps traveling down and forward to pass through the pelvic floor. And we call this portion of the urethra, if you remember, this is the intermediate or membranous urethra. This is a membranous or intermediate urethra. Then the urethra travels through the penis, if you remember. And we call this segment of the urethra, this is the penile or spongy urethra. This is how the penis is going to be allowing the urethra to travel on here. We divide the penis into 3 segments: root, shaft, and glands. So as you see, the urethra, the penile urethra or the spongy urethra is going to be getting into the penis through the root, traveling through the shaft of the penis, and then will be releasing the semen through the external opening of the urethra. If you remember, we call this as the external urethral orifice. So again, what did we mention so far? Seeing the testis on here, it's going to be kept in a skin pouch pending down from the pelvis outside of the body, and located above the testis we've got the epididymis that will be carrying the sperms from the testis to travel through a duct, and this duct is what we call the vas deferens or the ductus deferens. The vas deferens can carry the sperms all the way up to the pelvis until it reaches the base of the urinary bladder. This is where it's going to be its ampulla, its bulging part, will fuse with the seminal vesicle. This is a seminal vesicle on here. The blue, the one which is in blue color. The seminal vesicle duct, as it fuses with the vas deferens, they will be together forming another duct that we've called the ejaculatory duct. Right and left ejaculatory ducts going to be conducting the semen to the part of the urethra traveling inside the prostate gland, and we call this as the prostatic urethra. The urethra then travels to the outside of the prostate gland, passing through the muscles of the pelvic floor. We call this as the membranous or intermediate urethra. Then travels through the penis, passing through the root, shaft, and then glands. It's going to be releasing the semen out during ejaculation through the external urethral orifice. Another gland that will be associated with the male reproductive system on here is going to be a gland that's going to be releasing its secretions before ejaculation, and is going to be located at the entrance of the urethra in the penis. If you notice on here, the root of the penis has a rounded structure. This is called the bulb of the penis. Bulb of the penis. And those wings protruding out like this is going to be the crus. So you've got bulb and crus. So we're going to call the gland on here that joins the urethra as it travels through the bulb of the penis. We call it the bulbourethral gland. Bulbo because it's going to be joining the urethra at the bulb of the penis. Urethral because it joins the urethra, and it's a gland. So we call this as the bulbourethral gland. So again, how many glands did we see so far associated with the male reproductive system? We've seen first one is the seminal vesicle. Second one is the prostate gland. And the third one is going to be the bulbourethral gland. Another name for it is going to be Cooper's gland. Cooper's gland. So again, what we've mentioned so far, the gonad for the male reproductive system is going to be the testis. This is where the formation of the sperm is going to be taking place. Sperm is going to be conducted up into the epididymis. And then from the epididymis, the sperm is going to be traveling up all the way back to the pelvis through a duct that we call the vas deferens or the ductus deferens. Vas deferens is going to be fusing with the duct coming from the seminal vesicle, and together they will form another duct called the ejaculatory duct. Right and left ejaculatory ducts going to be releasing the semen to travel into the part of the urethra passing through the prostate gland. Again, we call this the prostatic urethra. The urethra then travels outside of the prostate gland, passing through the pelvic floor. We call this as the membranous or intermediate urethra. Then the urethra travels through the penis. We call this as my spongy urethra or penile urethra. Again, the penis is going to be divided into root, shaft, and glans. Root, shaft, and glans. If we're looking here at the root, you're going to have bulb and a crus. Bulb, penis, and crus. At the entrance of the urethra in the root of the penis, we've got a gland that releases its secretions before ejaculation. We call this the bulbourethral gland. Bulbourethral gland or Cooper's gland. The semen will be released from the urethra to the outside opening of the urethra, which we call the external urethral orifice. Three glands that we've seen associated here with the male reproductive system. First one was the seminal vesicle. Seminal vesicle. Then we've got the prostate glands that will be surrounding the neck of the bladder and the prostatic urethra. A third one, which is going to be located at the entrance of the urethra in the root of the penis. We call this the bulbourethral gland. Any difficulty? Any difficulty? Questions? Questions? This is one of the most important. This is definitely going to be a 10 stars diagram. This has to be on your exam. Has to be on your exam. Every single marking on it is going to be highly important for us. If you notice on here, associated with the male reproductive system, we can see some structures on here. We've got the pubic bone, and this is going to be the symphysis pubis, the joint that attaches the two pubic bones together. Located behind the prostate gland and the urinary bladder, you've got the rectum that opens up to the outside through the anal canal and anus. Let's look now at a cut section in the testis. So you can see here, inside the testis is going to be divided into lobules. And forming those lobules, you've got ducts. We call those are the seminephros tubules. nephros tubules. This is where the production of the sperm is going to be taking place. After I produce the sperm is going to be conducted to a network of ducts before they reach the epididymis. This network of ducts on here is what we call the rete testis. Then the sperm is going to be traveling to the ducts located inside the epididymis. We're going to be dividing the epididymis into 3 main parts. We've got head, body, and tail of the epididymis. So again, we divide the epididymis into 3 main parts: head, body, and tail. After that, the epididymis ducts will be conducting the sperms to travel into the vas deferens that will carry the sperms all the way up to the pelvis. If you notice on here, the vas deferens doesn't travel on its own. It travels accompanied by other structures like the pampiniform plexus of veins, the veins that are draining the testicular structures, and the arteries that will be testicular arteries that will be conducting the blood supply to the testes. All of the 3 gonna be traveling inside what we call the spermatic cord. Spermatic cord is the cord in which you're gonna have the vas deferens traveling, the veins, and the arteries. Right? If a person has a defect in the attachment of the testis to the surrounding, so a person might have a torsion of this. So what is the problem here with having torsion of the testis? What do you think is going to be the problem? Why you can't just rotate the testis around? Yeah, it's going to cut the blood supply. Exactly. It's going to cut the blood supply. So if it was not resolved very fast, what's going to happen? A person might lose this testis because it won't be functioning anymore. All right, so a person, in the emergency room, what they will do for him, they will untwist the testis, and most likely he will need to get it fixed by performing a surgery. So again, what we're looking at in here, we're looking at the testis, which is built by lobules, and those lobules are formed by seminephros tubules. This is where the spermatogenesis is going to be taking place. The seminephros tubules will conduct the sperms that will be produced through a network of ducts that will carry the sperms from the testis to the epididymis. We call this as the rete testis. Then I will be conducting those sperms to the ducts of the epididymis. Again, we divide the epididymis into 3 main parts. We've got the head, body and tail of the epididymis. From the epididymis, the sperm is going to be traveling through a duct in here. We call this the vas difference or the ductus difference. Ductus difference is going to be traveling all the way up towards the pelvis, passing through the spermatic cord, and it's going to be accompanied with the blood vessels and nerves, like the pampiniform plexus of veins, the spermatic arch, and so on, or the testicular arch. I'm sorry. Questions? All right. So if anybody is marking, please do not mark because everybody can see the marking, I believe. I'm not even sure how to erase it. So can you please erase the markings from the screen? All right, thank you. Any questions? We've got the coverings here of the testis. We've got the first covering that would be on the outside of the testis is going to be my Tunica Albuginea. And we've got another covering of the testis inside the scrotum. We call this double layered covering of the testis. This is the Tunica vaginalis. So again, two coverings that we can see on here. First one is going to be the Tunica albuginea. Second one is going to be my Tunica vaginalis, which is formed of two membranes. To the outside of them, you've got the scrotum, which is the skin pouch itself. So if we have a cut section of the penis like this, how does it look like? You're going to have two erectile tubes on the dorsum of the penis and one erectile tube on the ventral surface of the penis. This is how it's going to look like. All right, so again, looking here at the internal structure of the penis, what we can see two erectile tubes that are going to be located on the dorsum of the penis and one going to be located on the ventral surface of the penis. We call the two located on the dorsum, forming on the dorsum of the penis. Those are the corpora cavernosa. Corpora cavernosa. And the one that will be located on the ventral surface, this is where the urethra going to be traveling. We call this is the spongy or corpus spongiosum. Corpus spongiosum is going to be the erectile tube through which the spongy urethra going to be traveling. And if you remember, that's why we call the spongy urethra. So again, what we're looking at in here is a transverse section of the penis. The two dorsal erectile tubes on here, those are my corpora cavernosa, and the erectile tube on the ventral surface of the penis, this is going to be the corpus spongiosum. Corpus spongiosum. We're looking on here the different structures of the male reproductive system. Again, we've got the testis on here. Located just above it, we've got the epididymis. As a continuation of the epididymis, we form a duct that travels up towards the pelvis. We call this the vas deferens or the ductus deferens. The bulging part of the vas deferens is called the ampulla of the vas deferens. It will join the duct of the seminal vesicle and will form the ejaculatory duct. Right and left ejaculatory ducts going to be releasing the semen to the part of the urethra passing through the prostate gland. We call this the prostatic urethra. Then I have the membranous urethra. Then the urethra travels through the penis, penile urethra, to release the semen to the outside through the external urethral orifice. Three parts of the penis that you can see on here: root, shaft, and glands. The root, as you see, has bulb and crus. So this is the bulb of the penis, this is the crus of the penis. Bulb and crus. We've seen a gland located at the bulb that joins the urethra to release its secretions before ejaculation. We call this the bulbourethral gland. Bulbourethral gland. Questions? All right, great. So the penis is going to be the erectile organ in the male reproductive system. The spongy urethra and three cylindrical bodies of erectile tissue are forming the main structure of the penis. Two erectile tubes that will be located on the dorsum of the penis. Those are the corpora cavernosa, and the erectile tube, which is located on the ventral aspect of the penis, this is going to be the corpus spongiosum. And this is the one that's going to be surrounding the urethra. What is erection? Erection is simply, in one word, like inflammation, it's a vasodilation. You dilate the vessels, getting more blood delivered to the erectile tissue, and as more blood travels to the erectile tissue of the corpora cavernosa and the corpus spongiosum, this will enlarge the penis and allow it to become rigid, to allow intercourse to take place. So for the male reproductive system, those are the three very, very important diagrams for us. So I would say this is a 5 stars diagram, and this is going to be another 5 stars diagram. Again, every single structure on those diagrams is going to be of great importance for us. Accessory glands, we've got the seminal vesicles, which will be producing a viscous alkaline fluid, and its secretions is going to be will be representing 70% of the volume of the semen. Inside, as part of the seminal vesicle gland secretions, we've got fructose, we've got ascorbic acid, coagulating enzyme called vesiculase, and prostaglandins. Why do you think you've got fructose release? Why do you release sugar with the sperms? What do you think is the reason why I would be allowing fructose to travel with the sperms as part of the secretion? Yeah, energy. So I need a source of energy. So an easy source of energy here is going to be the fructose, which the sperms can break down to generate ATP. So this provides the energy needed for the sperms to be able to travel. The duct of the seminal vesicle, as we've seen, joins the vas deferens, and this will form the ejaculatory duct. What is the difference between the sperms and the semen? What do you think is the difference between sperms and semen? Exactly. So the sperms are the gametes. Those are the sex cells that have the ability to penetrate the ovum, that carry half of the genetic material of a diploid cell. Those are the sperms, the gametes, the reproductive cells, the sex cells. But the semen is simply the sperms plus all the glandular secretion. And the major part of those secretions is going to be coming from the seminal vesicle. 70% of the semen, of the volume of the semen, is going to be released from the seminal vesicles. Moving on to another gland, which is the prostate gland, that will be encircling the urethra. It's going to be located inferior to the bladder. It also surrounds the bladder neck. It's going to be releasing a milky, slightly acidic fluid that contains citrate, enzymes, and there is an antigen that we use as a tumor marker called the prostate specific antigen, or PSA. So if a person is worried about having a prostatic carcinoma, one of the things that we're going to be checking is going to be the PSA, which is used as a tumor marker. It's going to be responsible for the activation of the sperms. The secretions from the prostate glands, the prostate gland gonna be released to accompany the sperms in the part of the urethra that travels through the prostate gland. We call this the prostatic urethra, and this is gonna be during ejaculation. So while the ejaculation is taking place, the prostate gland gonna be releasing its secretions to join the sperms and the seminal vesicle secretions as part of the ejaculated fluid. A third gland that we've seen as part of the structure of the male reproductive system, remember, this is the one that joins the spongy urethra at the bulb of the penis, because this is the bulbourethral gland, or Corpus gland. It's a pea-sized gland that will be located inferior to the prostate. It's going to be releasing the thick, clear mucus that will be released prior to the ejaculation. So the ejaculated semen contains the prostatic secretions, the seminal vesicle secretions, as well as the sperms. This is the ejaculated fluid. But before ejaculation, prior to the ejaculation, there will be a thick, clear fluid that will be released from the bulbourethral gland, or the Corpus gland. And the main function here of this thick, clear mucus that gets released from the bulbourethral gland is to neutralize the acidic urine in the urethra. Of course, you know that the urethra is a common pathway for both semen and urine. So urine is acidic on here. It's going to be traveling down from the bladder through the urethra to be released during micturition. So if there was any remnants of this urine, in order for the sperms to be able to survive, I need to neutralize this urine acidity in the urethra. So who's going to be handling the urine acidity here is going to be the bulbourethral gland secretion. All right. Questions? All right, so if we're looking here, so I have a problem with acidity. So if I'm neutralizing the acidity in the urethra, how comes the prostate gland was releasing an acidic fluid that will be mixed with the sperms. So if the sperms do not like acids, and I did have to neutralize the acids in the urethra by the bulbourethral gland secretions, why would I be releasing acid as part of the prostate gland secretions? What do you think? But here, yes, cleaning, so I'm cleaning up the acids in the urethra by releasing this thick mucus. But here, why would I release an acidic fluid from the prostate gland? pH control. So what do you mean, Tyler? Can you explain more? So the way I look at this here, why would I be releasing acids if I don't want to have acids? It's simply looking at the acid as a stress. So think of it this way. So if I told you we have all remaining exams are gonna be due tonight. All right, so this is too much stress on you, right? So too much stress will let you do what? Give up. So you won't be completing the course. If I put a lot of stress on you, so you're gonna give up and you won't be completing the course. You drop the course. If I told you that all exams are due tonight, what if I told you that we won't have any exams until the end of the semester? Would any of you study anything? So you're gonna all get A's for this class and nobody will have to study anything. We don't have any exams. Would you study the material just for the sake of knowledge? Yes, yes. So all of you are saying yes. That's great. That's great. Even if we don't have any exams, would you study? Yes? No. Yeah, here comes the truth. No, no. All right. All right, so if I don't have any stress on me, would I be performing anything? If I don't have any exam, I won't be performing. I won't become active. So I need some stress, but not too much stress, because too much stress on the sperm will cause its deactivation. It won't work. But having no stress at all also will inactivate the sperm. So I need moderate amount of stress. So this is what we're trying to do here. If I am exposing the sperms to too much stress, I need to neutralize this stress. If I will be exposed to the urine acidity, I need to get it neutralized. But having a very neutral environment without any stress on the sperms, they won't become activated. That's why I will need to expose the sperms to some degree of stress, some degree of acidity for the sperms again to become activated. Does this make sense? Yes, it acts as a stimulus, as a stress. All right, so semen is only 2 to 5 milliliter of semen that will become ejaculated each time. So semen volume is going to be 2 to 5 milliliter that gets ejaculated per ejaculation. Each ejaculation going to contain around 20 to 150 million sperms per milliliter. So if 2 milliliter, this is going to be between 40 to 300 million sperms. And if you go to the high end, so it's going to be 5, it's going to be 100 to 150. 750 million sperms per ejaculation. All right, so again, what is the composition of the semen? Semen is, again, glandular secretions and sperms. Any questions? Any questions? All right, so we can have a 10 minutes break. We come back, discuss the gametogenesis. And this includes the spermatogenesis, and then we're going to move on to the female reproductive system after that. All right, after break, I will tell you exactly what we're going to be doing for the lab, the remaining two lab exams. All right, so we'll see you in 10 minutes. Any questions before we leave to the break? Any questions? All right, great. So we'll see you in 10. All right, so before we go back to the reproductive system, I want to have an announcement here. For the remaining lab exams, we still have 2 more lab exams. First one is going to be covering the blood and respiratory system, which going to be similar in structure to the ones that you already completed. You're going to have the multiple choice questions, which are diagram based, and you've got an essay question, which is the pathogenesis of the erythroblastosis fetalis. Like what we did have with the urinary system, we did have an essay question and the multiple choice questions, which are diagram based questions. This is going to be worth 50 points towards your course grade. The second lab exam is going to be covering the reproductive system and the digestive system. And this is going to be worth 100 points towards your final course grade. It's going to be same concept. It's going to be multiple choices, which are diagram based. You're going to be provided with a list of choices for each of the markings, and you're going to need to select the single best answer for those questions. Those are going to be around 90 questions, and you've got an essay question that we're going to discuss next class, which will be the oogenesis, folliculogenesis, and the menstrual cycle. All right, this is going to be the essay questions that we're going to have for the reproductive system, which is going to be out of 15 points. So this means a person can get up to 105 out of 100. So this is going to be your extra credit points towards this last lab exam. So recently, the college has adopted an automated proctoring system called Smarter Proctoring. It's the erythroblastosis fetalis that we discussed last class. This is erythroblastosis fetalis. So Smarter Proctoring is a platform on which you're going to be conducting those 2 remaining lab exams, as well as the optional final exam, which I will discuss more about later. So not everyone gonna be willing to attend the final exam. So I will ask you, for those who are gonna be attending the final exam, I will be discussing with you the structure of this final exam. For Smarter Proctoring, it's gonna be the platform on which we're gonna be conducting the exam. So the exam is gonna appear normally on Canvas, but it needs an access code. This access code is going to be provided through Smarter Proctoring. So what you're going to simply do, you're going to go to the proctoring platform, which you can see a new tab that did appear yesterday on Canvas, which says Smarter Proctoring, and you will be able to check the exam. You need to schedule the exam, and you need to have some of the technical requirements in order to conduct the test. What are the technical requirements for you to conduct the exam? You should have a webcam or, of course, a laptop that has a webcam. So you need to have a webcam that is fully functioning. You also need to download Google Chrome, because smarter proctoring is simply an extension that's gonna be attached to the Google Chrome. So you need to take the test, you open the proctoring platform through Google Chrome. You need to be plugged in throughout the length of the exam. You need also to make sure that you have a stable, reliable internet connection. At least 1 megabyte download speed and upload speed, at least 1 megabyte upload and download speed. And I believe this requirement is going to be satisfied by any of the providers here in Jacksonville. Make sure you are using a desktop, PC or a Mac, not a tablet, not a Chromebook. Make sure you are using a PC or a Mac, a complete desktop. A laptop is a laptop that has a full operating system is going to be a requirement. It's an automated proctoring. What this means? This simply means that it's going to be conducted by artificial intelligence. So, for example, the operating system, for example, if finds you looking around, or it did detect 2 voices in the room, or it did detect another face that appeared in the room, or it did detect that you have left the exam, it's going to be flagging this. And what I will do, what's going to be provided to me, is a recording, a video recording of you taking the exam, and the software is simply going to be detecting anything that might look that you are violating the exam integrity. So no kids in the room, definitely. Nobody in the room. Nobody in the room. You have to be looking at the diagram, at the webcam for all the exam time. You are taking an exam. All right. Make sure you have a separate room. Nobody can enter the room while you're taking the exam. Nothing on your desk except for your laptop or desktop that you're going to be conducting the exam through. So they will ask you to have a 360 examination of the room. So you're going to be moving before you start the exam, moving around, showing them that you have no notes, nothing is written on the wall. If, for example, a person kept looking at his hand, it's going to be flagged, and it will give you an announcement that states in order to complete the exam, you need to show your hands to the camera to make sure that you didn't write anything on your hand. Also, it's going to be taking control of the number of screens that you're going to have. So you can't have more than one display or the exam will stop. So you can't have the exam, for example, while this is my desktop on here, you can connect to another screen. You can't have another computer device while you're taking the test, which is turned on. The notification will pop up if you didn't respond, the exam will be canceled. It's not a notification. You won't be able to proceed with the exam until you show them your hands. All right, so what the system here will do is put a flag that you have been looking at your hands, looking away from the camera. So you're going to be responding to the notification in order to complete the exam. And as you proceed with the notifications, everything is going to be recorded and I will review. So for example, you looked at your hand, but you didn't, when you showed your hand to the camera, you didn't have anything on the hand. So I will neglect this flag and so on. Yes. Yes. Pop-up notifications, turn them off. You need to turn off the pop-up notifications. The pop-up blockers. How can you look at the camera and look at the screen at the same time? Because the camera is just above the screen. So while you're looking at the screen, you are directing. If you can, please borrow. If you don't have a device, please borrow one for the period of the exam, for those 2 times. But you can't use your phone on the exam. You can't use your phone. You can't complete the exam on the phone. There are many webcams available that you can purchase from any of the stores, like 20 bucks or something like this, from any store. All right, so having a webcam, I don't believe it's an issue, right? So webcam costs like 20 to 300, 400 bucks, like according. So you just need a basic webcam. You can't if it detects that you're looking at your phone or you have your phone near you, it will flag it and it might block the exam for you. So you don't need to have this. You don't want to have your phone ringing as if you're taking the exam in class. I'll deal with it as if you're going to be taking the exam in class. All right, so you don't bring your children in class. You don't have your phone turned on while you're taking the test in class. You don't look anywhere else other than the exam during the test if you're taking the exam in class. So think of it, you're conducting the exam in class. No, you don't have to pay for this one. This is sponsored by the college. That's why we've waited so long for us to not use ProctorU. Do you have to download the app? Yes, you will have to download the app. And in order for you to check out and make yourself comfortable using the smarter proctoring before the actual exam, I will be providing you with a sample quiz that is graded. I'm sorry, that is not graded. But if you didn't complete the sample quiz for me to make sure that you can use the operating system, if you didn't complete the sample quiz, I will not allow you to take the actual lab exams. If you did not complete the sample quiz, which is not graded, you won't be allowed to take the actual exams. All right? I don't want to have people taking on the actual exams. They tell me we've got problems, we couldn't complete. This will affect the exam integrity. Sample quiz is going to be available for you tomorrow. I will put an announcement on. I will tell you that it's available. And I will provide you with video instructions on how to register for the exam, how to schedule for the exam, and the whole procedure. So it's like a training. All right, so the sample quiz I will be providing to you, it's not going to be graded. It's not graded. But this is a requirement for you to take the exam. Nobody skipping this sample quiz will be able to take the exams. When they will be available, when are going to be the due dates for the 2 lab exams? Starting on Tuesday, this Tuesday, which is, let me double check here, what day of the week, what days? Starting from this Tuesday, which is going to be April 14th, you're going to have the exam available for you for 1 week. So it's going to be available from April 14th to April 21st. All right. So you need to schedule your exam anytime. Don't wait until the due date. Don't wait until the due date. You will see that the exam is going to be due on the 22nd, but it's not the actual due date. The actual due date is on the 21st because nobody is going to be allowed to take the exam after the 21st. This is for lab exam 3, which is covering, again, the blood and respiratory system. For lab exam 4, it's going to be available for you to complete starting from the 21st to April 28th. No, there will be no time restriction. I believe there is no time restriction because it's not a live proctoring. So there is no other person who is proctoring you taking the exam. So it's a automated proctoring platform. All right. So, but you need to schedule. You need to put the schedule. You need to put a specific time when you're gonna be making the exam. When you're gonna be taking the exam. All right. But this might be any time during the day. This is what I believe. All right. This is what I believe. This is what I did understand from the webinar I've attended last week. All right, so again, lab exam 4, which covers the digestion and reproduction, is going to be available for you to complete any time starting April 21st until the 28th. And again, 28th is not the actual due date. The actual due date that's going to be effective is the 27th. I need to have a buffer time here for me to be able to resolve any issues that might arise. All right? Questions? So again, this weekend, I will be providing you with the sample quiz. It's a few questions. It's not going to be graded. It's mainly for you to practice how you're going to be taking the actual exam. The sample quiz, I wasn't going to put like any questions like on the actual exam. I will ask you a couple of questions, general questions. Like I didn't decide actually what I will be putting on this exam. So all right, questions like, can you see this question? Please select number 1 from the choices, and I will provide you with some choices 1 through 10, for example, stuff like this, like to check the functionality rather than an actual material. No, you won't know the grades right away because of the essay portion that I need to review. The whole exam it's not about the proctoring. The proctoring has nothing to do with the exam itself. It has nothing to do with the grading. The proctoring is just recording you while you're taking the test, making sure that you are maintaining the integrity. It's going to be responsible to maintain the integrity of the exam, and that's it. And you can simply see the exam. You can see the actual exam on Canvas, but you won't have access to it because you don't have the code, the password to access it. All right, it's like taking an exam in the testing center. So you go to the testing center. The testing center has nothing to do with the test itself, the tested material, or the grading. All right, so the exam platform is still going to be Canvas. It's going to be almost identical to in terms of structure to the ones that you've seen for the urinary system. But the only difference is that it makes sure that you are not cheating on the test. Any questions? Yeah, you will need to download the app, and this is what you're going to be practicing using this simple quiz that you're going to be provided with by tomorrow. Right? It will tell you what are the exact steps, and it will tell you the name of the app. It will tell you everything that you need to know in the form of steps. Lecture exam 4 is going to be 60 questions. It's not available yet on Canvas for you. All right. It's not available yet on Canvas for you, but it's going to be 60 points, like the other lecture exams. The lab exam 4 is the one that's going to be out of 100 points. Lab exam 4, not lecture exam. All right. Lecture exam is not going to be proctored. No. It's going to remain like it is, like it was for the other lecture exams. All right. Back to the male reproductive system. If you remember, we did study before the break the structure of the male reproductive system. We've seen the gonad, the testis, located within a skin pouch, scrotum bending down out of the body. And remember, why is it located out of the body cavity, out of the pelvic cavity? To allow normal spermatogenesis, to allow normal production of the sperms. Because in order for me to have normal formation, normal production of sperms, spermatogenesis, I need to have a pressure, a temperature lower than the normal body temperature. So no, I can't produce sperms if the testis is kept in the pelvic cavity inside the body. So it's too hot for spermatogenesis to take place. Sperms, as they get formed from the testis, remember in those tubules, we call them, those are the seminiferous tubules. We're going to be conducting the sperms to the rete testis, and then to the epididymis, which is located just above the testis. And then from the epididymis, the sperm is going to be traveling up through a duct. We call this as the vas deferens. And the vas deferens carry the sperms all the way up to the pelvis and will be meeting with the seminal vesicle located underneath the urinary bladder. Remember, the fusion between the ampulla of the vas deferens and the seminal vesicle duct is what will form the ejaculatory duct. Right and left ejaculatory ducts are going to be releasing the semen to the part of the urethra passing through the prostate gland, traveling through the prostate gland. And then the urethra, as it travels out from the prostate gland, is going to be called the intermediate or membranous urethra, and then will pass through the penis, because this portion of the urethra is the penile or spongy urethra. At the entrance, we've got the gland on here that will be releasing this clear, thick mucus that gets released prior to ejaculation to neutralize the urine acidity in the urethra. So this gland is what we call the bulbourethral gland. We did look at the structure of the penis from inside. Remember, there is 3 erectile tubes, 2 on the dorsum of the penis. Those are the corpora cavernosa, and one on the ventral surface of the penis is going to be the corpus spongiosum. So again, three erectile tubes. Those are the two corpora cavernosa and the corpus spongiosum. All right. So now we're starting the gametogenesis. What's gametogenesis? From the name here, Genesis means the formation, creation, and this is the formation of the gametes, the sex cells, sperms and eggs. Most of the body cells are diploid. What I mean by diploids, I have a complete number of chromosomes. We call this as a diploid cell or 2N. They contain two sets of chromosomes. One set of the chromosomes is going to be the maternal chromosomes, and the other set of the chromosomes is going to be the paternal chromosomes. So all of your cells will have two sets of chromosomes, a set coming from your mother, and the other set is coming from your father. So you've got 23 chromosomes from your father, 23 chromosomes from your mother. This gives you a total of 23 pairs of chromosomes that are going to be present in the nuclei of all your body cells. What makes the gametes different? Gametes are going to be haploid. What I mean by haploid cell, haploid means they only contain half of the genetic material, just one set of the chromosomes. They only gonna be containing 23 chromosomes, not 23 pairs of chromosomes. In order to understand how your body is gonna be producing gametes, we need to understand a type of cell division called meiosis or meiosis. Meiosis is going to be two consecutive cell divisions preceded by only one replication of the DNA. So if you remember, we did discuss the mitotic division. And if you remember, mitosis is the nuclear division. If I'm starting with 2N, so what happens before the cell divides? You're going to be duplicating the genetic material and performing one cell division. So each of the daughter cells going to have a complete set of chromosomes. I am producing two identical cells that look like the mother cell. This is what we call mitosis. But meiosis here I have only one replication of the genetic material during the process and two successive cell divisions. So what this results in, I'm getting 4 cells. Each of them gonna have only half of the genetic material. So again, what is the difference here between mitosis and meiosis? is going to be the number of cell divisions. How many times did you duplicate the genetic material for either a mitotic division or a meiotic division? You have only one duplication of the genetic material. The difference here is the number of cell divisions that you're going to be performing. In a mitotic division, I am performing only one cell division after one duplication of the genetic material. Here for meiosis, I will be performing one duplication of the genetic material, which is going to be followed by 2 cell divisions. All right. Any questions? Any questions so far? All right, so let's look back at the mitosis. How mitosis did take place. If you remember, the cell has a life cycle. We call the life cycle of the cell is the, you remember? The life cycle of the cell. Something so easy. Cell cycle. And remember, the cell cycle was divided into 2 main parts. We've got, most of the time, the cell is going to be in interphase. For most of the time. And there is time when the cell going to be dividing itself. This is what we call the mitotic phase. And if you remember, the mitotic phase was subdivided into time when the cell is performing the nuclear division. I'm dividing the nucleus into 2 nuclei. We call this as mitosis. And the time when the cell is going to be splitting those 2 nuclei apart to form 2 daughter cells, and we call this as cytokinesis. For the interphase, we did divide the interphase into 3 phases. We've got G1, we've got G2, and the S phase. G1 and G2, this is when the cell is growing. So I'm growing, I'm increasing the amount of cytoplasm, I'm growing the number of organelles, I'm growing the number of ribosomes, everything, doubling the number of tubules that will be used as Golgi apparatus, rough endoplasmic reticulum, smooth endoplasmic reticulum, everything, it's double. Cytoskeleton. This takes place during G1 and G2 phases of the interphase. During S phase, what's going to be doubled is going to be the genetic material. I'm doubling the number of chromosomes. It's DNA. DNA replicates, exactly. So if you remember, the cell cycle has, again, 2 major parts, interphase and mitotic phase. So what we're focusing on is the mitotic division. If you remember how the cell looks like during interphase before I start the cell division. If this is my cell like this, and this is the nucleus. If you notice, the nucleus has a nuclear membrane. It has its genetic material in a fluid form. It's called the chromatin. And I have a nucleolus. And I have the centrosomes formed of centrioles, like this. So this is how the cell looks like during interphase, before and after it divides. When I start the mitosis, what will happen? During the first phases of the mitosis, prophase, the nuclear membrane disappears, the nucleolus disappears, and the genetic material is going to be condensed in the form of chromosomes. Each chromosome is formed of two sister chromatids attached at the center. And the attachment on here is called the centromere. Also, you're going to have the mitotic spindle that will be formed, and they will be attaching themselves to the chromosomes from those centrioles, like this. What happens next? The mitotic spindle fibers that you see on here will be pulling the chromosomes, and as a result, the chromosome is going to be arranged at the equator of the cell, like this, as you have pulled them from both sides by the mitotic spindle fibers. We call this phase is my metaphase. You kept pulling the two sister chromatids away from one another, moving them towards the two poles of the cell. So they will be split apart, and we call the phase in which this takes place, it's called anaphase. So if I'm looking at the cell during anaphase, how does it look like? You have the sister chromatids have been separated from one another, and they are pulled towards the two poles of the cell. And as they are pulled from the center, they will have this V-shaped appearance. So now I have formed two sets of what? Would I call this a chromatid, or is it a chromosome now? What do you think? Anybody still with me? So how would I consider this as a chromosome? And this whole thing also as a chromosome. Although this one is half of this one, this chromosome was formed of two sister chromatids that I've separated them. So this one should be a chromatid. Why would I call it a chromosome? All right, so if I have a book on here, this is book one. I have two copies of this book. So how many books do I have? What do you think? In terms of information, I have the same exact book, but I have two copies of the information. So this simply means I, in terms of information, have one book. I have one chromosome that has this information, but twice. So if I give you one of those copies, so how many books would I have in terms of information, and how many books would you have in terms of information? Each one of us would have a complete set of information that is provided by this book. You see on here? So having two copies of the book present on two sister chromatids, this means I have the information coded on one chromosome. So we call it a chromosome. When I separate the two copies from one another and provide each cell with a copy, each of the daughter cells with a copy, so this means that each of the two cells that will be formed will have the same exact amount of genetic information like the mother cell, and they can make copies of this information as they want. So the chromosome, look at the chromosome as the amount of information that you have, the genetic information here. So if I have a genetic information, it's a chromosome, regardless of the number of copies here. So I have two copies of the chromosome, but we don't call it a chromosome because we don't call each of them as a chromosome. We call them, as they are attached to one another, those are sister chromatids. Does this make sense? So here I have 23, let's say, pairs. Here I have 23 pairs for each of them. No reduction. So if I separate the two chromatids, each one of them is going to be a chromosome. It has a complete set of information. So it's not required to have two copies of the book to have the knowledge in the book. You see what I mean? You don't have to have two copies of the same textbook in order for you to gain the knowledge in the textbook. You just need to have one copy to get all the information that you want. So if you have all the information, you've got the chromosome. Does this make sense, Joseph? So when they are connected, we don't call each of them as a separate chromosome. We call each of them as a chromatid. Once they are both attached, the two copies are attached to one another. So we call them two copies of the book. Yeah, each of them has a complete set of information. Each of them has a complete set of information provided on this specific chromosome. All right. So I split the chromosomes away from one another, forming two sets of chromosomes. And this takes me to the last step in the mitotic division, in which I will be reforming the nuclear membrane around those chromosomes. The chromosomes will turn into chromatin. The nucleolus reappears, and the mitotic spindle fibers gonna disappear. And we called this phase, if you remember, this is my telophase. During Anaphase, and during telophase, and after telophase, you can keep going, forming this constriction ring to split the cell into two daughter cells. We call this process is cytokinesis. That means the cell division. All right. This is an overview of the mitosis that we've studied back in AP1. Any questions? Any questions? So you start here with a diploid cell, 2N, and you're gonna get two daughter cells, each of them, sure. Each of them has the same number of chromosomes like the mother cell. Each one has 2N, complete set of chromosomes. If we're looking at meiosis, the chromosomes are not going to be arranged like this that we've seen in the mitotic division, singles, but they are arranged in pairs. You've got a paternal chromosome, let's say chromosome number 1 from your father is paired with chromosome number 1 from your mother. Chromosome number 2 from your father is paired with chromosome number 2 from your mother, and so on. They will be arranged in pairs. So it starts with a chromatin like this, and then the chromatin gonna be condensed. The centrioles are gonna be paired, and each centriole is gonna be traveling to a pole of the cell, forming the mitotic spindle fibers on here. a major difference that you see on here is that those chromosomes that will be formed are going to be arranged in pairs, not singles. This is during the first cell division of meiosis. This is meiosis 1. Remember, we've got 2 successive cell divisions that will be taking place. 2 successive cell divisions. We're looking here as the first one, meiosis 1. Centrioles are going to be attaching themselves with the mitotic spindle fibers to the chromosomes. What do you notice on here? The chromosomes that are paired together will start to exchange genes. So some of the blue genes did move to the yellow chromosome, some of the yellow genes did move to the blue chromosome. Then those paired chromosomes gonna be separated and will be moving away from one another to the 2 poles of the cell. This will be followed by the formation of the constriction ring on here, and cytokinesis will be taking place, separating them from one another. So I started by having 2N. This means what? I have chromosomes in pairs. I have 23 pairs of chromosomes. I have 23 pairs of chromosomes. I have separated the pairs from one another. So here, how many chromosomes each will have? What do you think? How many chromosomes? 23 only, not paired. But didn't I duplicate the genetic material? What do you think? Didn't you duplicate the genetic material? And when you perform a cell division, how come you're gonna have 23 chromosomes? If I did already duplicate the genetic material, how come would I have 23 chromosomes in each of them? Any idea? Any idea? So remember, you have how many books in your cells? How many books do you have in your cells? You've got 46 books. You've got 23 from your father. You've got 23 from your mother. When you duplicate the genetic material, this means what? This means that I did form another copy of those chromosomes, of those books. So now I have 46 or 23 pairs, but not only pairs, I have 2 copies of each. So what I was doing here in the mitotic division, I give 1 copy for each of my daughter cells. I've got 2 daughters, and I would be given each of them a copy of the whole 23 pairs. So this one got 23 pairs, meaning 46 different chromosomes, 46 different books. This one got 23 pairs, 46 different books. But here, in this case, I've given this daughter cell 2 copies of those 23. And the other daughter cell is gonna get 23 books which are different from those ones, those are not the same. Those are not the same. But the daughter cell here also gets 2 copies of those 23 books. So how many books, in terms of information, each of them will get? So here, this one got 23 books, this one got 23 books. But each of them got 2 copies of those 23 books. So how many books, in terms of information, each of them get? 46, 46, 46, 2, 23. Any other ideas? $46. So again, you have a library, you have 23 books. Of history. And 23 books of biology. You make a copy of each of those books. So I've made, now I have 2 copies of the 23 books of history. I have 2 copies of the 23 books of biology. And I give one of my children all the 2 copies of the 23 books of history. And my other child, I have given him 2 copies of the 23 books of biology. In terms of information, how many books the first child here has? In terms of knowledge. And the second one, in terms of knowledge, how many books this second child has? 23. Exactly. This is exactly what happened in here. Yes, you did duplicate the genetic material. You have 2 copies of the 46 chromosomes, 23 books of history, 23 books of biology, but you have 2 copies of each one of them. But when you split the cell, you're going to give 2 copies of the 23 books of history to one cell and 2 copies of the 23 books of biology to the other daughter cell. So each of them has half of the information. I have half of the information that the mother cell did have. Does it make sense? So this child has all the biology, this child has all the history, but the mother cell did have both the history and the biology. It had all the information. So here, each of the daughter cells gonna have half of the information. Does this mean I am a haploid or a diploid cell? What do you think? Am I a haploid or a diploid cell? Remember, what's a haploid cell? Haploid cell means I have half of the genetic material. A diploid cell means I have a complete set of chromosomes, have the number of chromosomes. Here, in terms of chromosomes, each of them has half the number of chromosomes. Remember, chromosome is all about information, genetic information. So here I have half of the genetic information. Here, and the other child also has half of the genetic information. Make sense? Make sense? What happens next? You're going to be performing the second cell division, which is going to be almost identical to the mitotic division. You've got 23 books on here, but 2 copies. Here you have 2 copies of 23 different books. So what will you do? You're going to be passing through the same steps. You're going to pass through prophase, metaphase. You arrange the chromosomes at the equator of the cell. You split the chromosomes apart, forming 2 sets of chromosomes that will be surrounded by the nuclear membrane in the telophase and cytokinesis. So what we're going to be doing, we're going to be given each of the grandchildren on here 23 chromosomes. Remember, each of those chromosomes did have 2 copies of the same book. So let's say this is history book number 1. I have 2 copies of it. So I give 1 copy to this child, 1 copy to the other child. So how many cells would I be creating? I'm creating 4 cells. Each of them has only half of the genetic material, only half the number of chromosomes. Questions? So I'm producing here 4 haploid cells. Looking at spermatogenesis. Spermatogenesis, from the name, it's the formation or the production of sperms. I have 3 phases. Mitosis, mitotic phase, in which the stem cell responsible for spermatogenesis gonna be perform a cell division. We call the stem cell here is spermatogonia. Spermatogonia gonna be performing a mitotic division, mitosis. So again, can you remind me what's mitosis? I'm producing how many cells? And are those diploid or haploid cells? You're producing 2 daughter cells, which are diploid or haploid, 2 diploid cells, exactly. So 2 daughter cells are gonna be formed. Each one has a complete set of chromosomes. This step is the reason why males don't have menopause. Why? Because you see on here, those are 2 daughter cells that are similar to the mother cell. So one of them will be reused as a stem cell again and again and again. Compared to in the female reproductive system, this step only takes place during fetal life, before birth. Like the brain cells, can they divide? They did divide to form the brain, but after birth, they are not dividing anymore. So the same concept on here. So in the female reproductive system, the mitotic division of the stem cells only takes place during fetal life. It stops after birth. So I will have a limited number here of those premature cells. Those premature cells, once consumed, this will result in menopause. So if every month after puberty, I'm consuming some of the reserved eggs. And as they get depleted over time, this simply gonna result in menopause. Why again menopause won't take place in males? Simply because of this step, mitotic division, that takes place for the spermatogonia. Spermatogonia on here, once divided by mitosis, it's gonna be forming spermatocytes. Spermatocyte can become spermatogonia to split again and keeps producing sperms. Spermatocytes will divide by meiosis. To produce, how many cells do you think are going to be produced from a meiotic division? 4 cells. And are those cells haploid or diploid cells? Each one only going to have half of the genetic material. They are haploid cells. And we call those cells, those are the spermatids. So if you remember, how many cell divisions will be taking place in meiosis? 1 or 2 cell divisions do I have? I have 2 cell divisions. I have meiosis 1, which is going to be followed by meiosis 2. Meiosis 1, we call those are the primary spermatocytes that will split in meiosis 1 into 2 haploid cells, which are my secondary spermatocytes. That will then complete meiosis 2 to produce the spermatids. Are the spermatids the actual gamete? What do you think? Can those spherical cells that are produced, can they travel through the seminiferous tubules, then retitestis, then the epididymis, then the vas deferens, then the ejaculatory duct, prostatic urethra, membranous urethra, spongy urethra, and then will be ejaculated through the external urethral orifice to the vagina, to the cervix, internal os, external os, cervical canal to the endometrial cavity to the fallopian tube. Are those spherical cells can move like this? Would they be able to swim all this, travel all this distance for them to fertilize an egg? There is no way, no way. So I need to modify the shape. So nothing gonna change in the genetic composition. So I still gonna have this same genetic composition. I have only half of the genetic material. I'm only gonna have half of the genetic material, have only n number of chromosomes. But I need to change the shape, not the composition of the genetic material. So what I have in here is the modification of the shape. So I will have a middle piece on here of the sperms that contains a lot of mitochondria. The sperm gonna have a pointed apex that contains hydrolytic enzymes, digestive enzymes to allow it to penetrate. It's gonna have a tail with contractile units that allows it to move and allows the propulsion of the sperm forward. And inside the sperm, you still gonna have the same number of chromosomes. Nothing gonna be changed in terms of the genetic composition between the spermatid and the sperm. The process in which you're gonna be modifying the spermatids into sperms, this is what we call spermiogenesis. Please notice the difference here between spermiogenesis and spermatogenesis. What is spermiogenesis? This is the modification of the shape of the spermatid into a sperm. The spermatogenesis includes what? Includes mitosis, meiosis, and spermiogenesis. All the three are steps in spermatogenesis. Spermiogenesis is this final step in which you're going to be modifying the spermatid into its final shape, which is the sperm. Is this clear? Clear? Any questions? All right, great. So if we're looking inside the seminiferous tubules, we will see cells that are playing a major role in providing nutrients for the growing sperms. They are disposing any excess cytoplasm that sloughs during spermiogenesis. When you're cutting the spermatid and modifying its shape, you're going to be sloughing the extra cytoplasm to get this final shape of the sperm. So I have cells that will be taking away this sloughed cytoplasm. It's also going to be producing the fluid in the testis in which the sperm is going to be traveling. So if you remember, the sperms are traveling here. Seminiferous tubules to the rete testis, to the epididymis, and then to the vas deferens. All this before I receive any secretions from the seminal vesicle. So how the sperms would be able to travel all this distance before I actually receive the seminal vesicle secretions. Simply because the testis itself is going to be producing fluid, testicular fluid, in which the sperm is going to be traveling to reach all the way up to the ejaculatory duct before, or close to the ejaculatory duct, before you receive the glandular secretions from the seminal vesicle. All right, so what is going to be the name of the cell on here that is responsible for all this? Those are called my Sertoli cells. Another name for Sertoli cells, those are my sustentacular cells. So again, what are the Sertoli cells? Those are cells within the seminiferous tubules and in the testis, and they would be responsible to provide nutrients to the growing cells and provide signals for the cell division to take place. Also, they will be responsible to dispose any excess cytoplasm that gets sloughed off during spermiogenesis, and they will be responsible for the testicular fluid formation that allows the sperms to be transported up through the seminiferous tubules, retitestis, epididymis, and vas deferens. So we're looking on here again at the steps of spermatogenesis. You've got the spermatogonia, which is my stem cell for spermatogenesis. You're going to be performing a mitotic division, giving rise to a cell that finds its way to complete the spermatogenesis, and another cell that might be used as a stem cell another time. We call the one that proceeds in spermatogenesis, this is a primary spermatocyte. Primary spermatocyte is going to be passing through the first cell division, which is meiosis 1. And meiosis 1 will allow you to form two daughter cells. Those are called the secondary spermatocytes. Each of them gonna be dividing into two daughter cells. Those cells are called my spermatids after I pass through the second division of the meiosis. Again, those spherical cells cannot travel through the sperm carrying ducts, through the female reproductive tract. So I need to modify the shape of those cells. So again, the process by which you're going to be modifying the shape of the spermatid into a sperm is what we call the spermiogenesis. Spermiogenesis. The whole process is what we call spermatogenesis. Any questions? Any questions?